How do our cells repair genetic mutations? Have you ever wondered how our cells fix the mistakes in our genetic code? Genetic mutations can happen for many reasons, like exposure to sunlight or even just normal processes inside our bodies. Thankfully, our cells have developed clever ways to repair these mutations and keep our genetic information intact. Let's break down how this works. First, cells have a system to detect when something goes wrong with their DNA. Special proteins act like sensors, looking for any irregularities in the DNA structure. These sensors can spot mismatched bases, breaks, or chemical changes. Once they find damage, they send signals that pause the cell cycle. This pause gives the cell time to fix the issue before it copies its DNA or divides. There are several main pathways that our cells use to repair DNA damage, each designed for specific types of problems. One of these is called mismatch repair. This system corrects small errors made during DNA replication, like when bases are paired incorrectly. It identifies the new strand of DNA, removes the wrong base, and replaces it with the correct one. If there are issues with the genes responsible for this repair, it can lead to conditions like Lynch syndrome, which raises the risk of colorectal cancer. Another pathway is base excision repair. This method fixes small damage caused by oxidation or other factors. Enzymes called DNA glycosylase recognize and remove the damaged bases. After that, they cut the DNA backbone and fill in the gap with the right nucleotide. Nucleotide excision repair is another important pathway. It deals with larger DNA lesions that distort the DNA structure, such as those caused by ultraviolet light. This pathway removes a short segment of DNA containing the damage and uses the undamaged strand as a guide to fill in the gap. Double-strand breaks are particularly serious. Cells repair these breaks using two main methods. Homologous recombination uses a sister chromatid as a template to fix the break accurately. This process mainly happens during the S and G2 phases of the cell cycle. The second method, non-homologous end joining, directly connects broken ends without a template. While this method is quicker, it can introduce errors. Repairing DNA also requires changes to chromatin, which is the combination of DNA and proteins. Proteins like poly-ADP-ribose, polymerase I quickly gather at the breaks and signal for repair machinery to come in. If the damage is too severe and cannot be repaired, cells may choose to undergo programmed cell death, known as apoptosis, or enter a state called senescence, where they stop dividing. These responses help prevent the spread of mutations that could lead to hereditary health issues or cancer. Understanding how these repair mechanisms work is important, especially in the context of family genetic testing and hereditary health. Mutations in the genes responsible for DNA repair can be passed down, increasing the risk of certain cancers and genetic disorders in families. Genetic testing can help identify these mutations, allowing individuals and healthcare providers to make informed choices about monitoring and treatment. In summary, our cells are constantly working to repair genetic mutations through various specialized pathways. These processes are essential for maintaining genetic stability and play a significant role in hereditary health and family genetic testing.